Howdy folks, and welcome to this microbiology video where we're going to be talking about how to do an endospore stain. Now if you remember back from lecture, an endospore is when a bacteria pulls itself together in an instance of drought when nutrients are scarce so that it can try to survive until the next time nutrients are once again available. Not all bacterial species are able to form endospores, so what this test does is reveal with a differential stain which bacterial species is actually able to form an endospore. Now, what you're going to notice when you come into lab is that you have two different sets of tubes available. You have two different bacterial species that have been cultured for 1 day on a slant, and those same two different bacterial species cultured for 4 days on a slant. The reason for that is 1 day on a slant is not enough time for an endospore forming species to form endospores. However, on a 4-day culture on a slant, an endospore forming species of bacteria should start forming endospores. Okay? So what you're going to be doing is working together with your lab partner. One lab partner will be performing the stains on the 1-day cultures, and the other lab partner will be performing the stains on the 4-day cultures. And then you're going to want to work together when you're visualizing your stains at the end to determine which bacterial species is the endospore forming species. Okay? So, to start, what we're going to do is get our slides out, and the first thing we always want to do is label our slides. That way we know which side is up and where we're putting our different cultures. Now, what I'm doing is I'm labeling them, each side of the slide, unique for which culture I'm going to be smearing on that respective side of the slide. The reason for that is we're putting both smears here, so you don't want to mix up which smear contains the bacteria that has the potential endospores. Okay? So get them both labeled very clearly so you know what you're working with. And then, because we're coming from a slant, the first thing we are going to want to do is to get water onto the slide for the formation of our smear, because these are solid cultures, just like when you pulled from the agar plate. You need to use sterile water to serve as the mixer. So get a little bit of water. We've got a little bit of water there now. 2 to 3 loopfuls of water should be good. I'm then going to read my label, find the appropriate culture, sterilize my needle. And again, the important reminder here is that you do not need to scrape off culture. Just barely kiss the edge of where you see bacterial growth. You do not need a ton for this. Okay, so let it cool. Flame the mouth of the tube. Find the edge of the bacterial culture. And before you touch it, go ahead and touch the side of the tube just carefully. Make sure your needle is cool, and then kiss the very edge of that culture. Bring it out. Flame the mouth of the tube. Recap. And then take that little bit of culture and dab it into the water and shake it around a little bit to get it free, and then sterilize. Set it down to let it cool, and get your already sterile loop, because you've already sterilized it after getting new water. Take that to spread your smear out. Get those bacteria spread out. And sterilize. And we're going to repeat this for the other culture. Okay, so let your loop cool. Remember always keep your the tools that you can near the flame because it creates that zone of inhibition, reduce potential contamination. Once it cools, we can then collect our water. Again, 2, maybe 3 loopfuls, depending on how much you get transferred to your slide itself. Sterilize and let it cool. Remember, we've already sterilized this. As long as it hasn't touched anything, we're good to go. Check your label for the side of the slide you're on and the culture you're using. Flame the mouth. Just kiss the edge of that culture. Flame the mouth. Cap. And then drop that little bit of culture into that droplet of water. Sterilize. Set it down to cool. And take your sterile and cooled inoculation loop to spread that droplet of water around to create your smear. All right? And again, sterilize this because you just touched bacteria in that water droplet. Set it down to cool. Now, while we're waiting for those to air dry, we're going to get our microwave apparatus ready. So you're going to have one sheet of paper towel, fold it over twice to form a nice little square. Stuff it down into this glass bowl. Take your water and spray it to soak that paper towel. Now you want to spray generously so that the whole thing gets soaked, but don't spray over generously so that your paper towel is swimming. You want to make sure it's completely covered. All right? Now, while this is air drying, what we're going to do, let me finish explaining, you're going to then, once this is ready to go, and this is heat fixed, we're going to put our slide in here, add our malachite green stain to it, and then we're going to microwave it. Now, why do we microwave it? The heat from the microwave will function as a mordant. Where have you heard that term before? We've used that term for iodine in gram stains. The mordant helps lock the color in. In this case, the heat is functioning as a mordant to help get that malachite green color into the endospore forming bacteria, and it'll lock that stain into the endospore structure, okay? So, this is now air dried. Be careful not to burn yourself. Pass it over. 1, 2, 3, 4, 5. And then once heat fixed, simply set it down into your bowl, and you want even distribution of heat with your slide. So the critical thing is to make sure you come in and fill in underneath the slide with water. You want full contact with the bottom of this slide. Notice the air bubbles will go away. If you do it just right. There we go. As long as there's not an air bubble under where you made your smear, you're golden. Okay? So, then we take our malachite green, cover where we have the smears, but you don't want to overcover because you don't want that malachite green to get down on the paper towel. You want it to stay directly on the smears. Close the die, and carefully, not to jostle the malachite green off the slide, bring it over here to the microwave and microwave this for 30 seconds. So just a reminder, the process of microwaving this is we're using the heat generated here to help drive that dye deeper into the endospore so that then it is trapped within that endospore structure. Without applying heat in this manner, that dye is going to simply sit on the outside of the endospore structure, and when we wash it off here in a moment, it'll all wash away and we'll have nothing staining the endospore. All right, so now we have this lovely mess. Bring it back over here. And if you don't want to get your hands dyed, use gloves. Pull your slide out, set it down here, and remove your paper towel as waste so that then your lab partner or someone else waiting on one of these glass bowls can utilize it. Now take your slide, and we're simply going to wash malachite green off, just like you wash off any other dyes. Be very careful not to forcibly wash off your bacteria.
Probably want to wash it a fairly long time.
Yes. And I'm coming down here below my my B sub smear to then make sure I get the dye off the E. coli smear. You want to wash until this washes clean, until you see no dye running across the smears. Dump here one more time. Just be careful not to forcefully spray the bacterial smear itself. Okay. Now, as long as an endospore is formed, that malachite green that we microwaved into the endospore has remained here. All other malachite green should have washed off of the bacteria. If that's the case, then when we come in to counterstain with safranin, all of those cells should pick up the red dye. So we're simply going to add safranin to the spots where we know our smears are. And we'll set a 1 minute timer. Don't forget to have this ready. Let the safranin sit for 1 minute, and the safranin is acting as a counter stain. So while we're waiting for this 1 minute with the safranin dye, let's just cover what you're going to see. If it is a fully formed endospore, you're only going to see green because the heat we applied to the malachite green drove that into the endospore structure, and so it'll show up green. If it is a bacteria that is in the process of forming the endospore, you will see both red and green. Why? The endospore structure itself will soak up the green, but all the other part of the cell will remain red, and then any cell that isn't forming an endospore will show up entirely red because there was no endospore to soak up the malachite green when we applied the heat. All right, so our minute has passed. We will simply wash this off. And dab dry with bibulous paper. Again, be careful not to rub the culture, the smear off of the slide. I still see a lot of people rubbing the paper. That's a no-no. You risk wiping the bacteria off. And now we have an endospore stained slide.